cases of people with brain tumors. Two were primary, two were secondary, and one primary. All right. Right. Generally, brain tumors, we can classify them into two areas. Tumors that grow in the brain and brain tissues like the nerves, blood vessel, the glial cells, or sometimes they call it nerve cells, that provide nourishment to the nerve. They are called glioma, right? In adults. In children, they are called medulloblastoma. Of course, adult. Sorry. This glioma can be also another word like I mentioned later. Is glioblastoma. All right. The other tumor is tumor around the membranes of the brain, which is surrounding the skull. They are called meningioma. All right. Primary brain tumors generally start off, doctors will always tell you it's benign because it's slow growing and it doesn't invade nearby tissue, right? Only when it gone into very advanced stage that they will say it is malignant. So it can stay benign for maybe 7-10 years, that kind of long time, right? So most of the time, doctors, if you have a little bit of a problem, you go to a doctor. Doctor will always say, "Oh, it's benign. We will observe and see." All right. Page 2. Okay, symptoms. Next slide. All right, symptoms of brain tumors depending on the location and the size of the tumors. All right. So usually when you press on a certain nerve area, like people always consider headache, giddy, limb pain, a lot of neurological functions thing. It's very common. All right. So I did not go into detail on what exactly it is. Those who want to know more about it can go and read about it. All right. The incidence of primary brain tumor is only a quarter of all metastatic brain tumor. All right, and malignant brain tumor account for only 1 to 2% of all malignant tumors. So let's say 100% of the cancer, probably only 1 or 2 are brain tumor. All right. But treatment of brain tumors are the most expensive of all cancer. Right? This therefore highlight the disproportion between the hope of the people when they go for cancer treatment and the low survivability of the patient and the cost involved. Right? I mean, you let's say the treatment you go, maybe it costs 30,000, 40,000, but the survivability is very low. Right? So that's why the next paragraph says that despite current multimodal therapies, right? Therapies like surgery, radiation therapy, chemotherapy, the prognosis of patients with this tumor, brain tumor, remains poor. Overall, the survival of glioblastoma patient, glioma, just in short, glioma, only at 2 years is only 27%, a quarter percent. So even with new aggressive standard of care, special care, all that thing, he can only live not more than 2 years, and it's only half a quarter of them. All right, the brain as part of the nervous system is unique because of the blood-brain barrier, which prevents most drugs from reaching it. Okay, so that explains for why is very expensive and very hard to treat. Okay, so adjuvant immunotherapy to improve a patient's immune system has been the focus of brain tumor treatment in recent years. Why do the medical fraternity look into this immunotherapy treatment for brain cancer? Is because of this blood-brain barrier. All right. So blood-brain barrier, you use a body system, the immune system to fight against the cancer, right? Which is at least lesser resistant than drug, which the body prevent it. The blood-brain barrier, the triple B, prevent it from reaching the cancer. Okay. All right. Let's next slide. Okay, Lingzhi has been, as you know, Lingzhi has been widely used in East Asia for general health treatment and longevity, right? And for the treatment of various human diseases for over thousands of years, right? 4,000 over years. Right, the bioactive compounds found in Lingzhi like polysaccharide, terpenoid, and protein have been found in recent research to have immunomodulatory and anti-tumor effects. All right. Over the years, if you have been following these scientific studies, you know that Lingzhi compounds have been having this anti-tumor effect. So therefore, this they found that that is the case. So since that is the case, why not the researcher look into it to treat use this process to treat brain cancer, the immunotherapy process that they have been looking at. All right. So in this report here, they feature 3 research reports that shows the inhibitory potential of Lingzhi and on malignant glioma and meningioma. All right. These 3 reports were anti-tumor and immunomodulatory activities of Ganoderma polysaccharide in glioma-bearing rats. 2, Ganoderma microsporum protein suppresses cell mobility and increases temozolomide sensitivity. This temozolomide is a chemo drug through induction of slug degradation in glioblastoma multiforme. Right. The third research report was talking about ganoderma acid, which is a terpenoid. A and DM induce NDRG2 overexpression that suppresses high-grade meningioma growth. All right. These are the heading, the title. So if you want to read a little bit more, maybe you can read the abstract. It's available free. But I think if it is the detail might be a bit difficult. Okay, let's look at the next slides. Okay, generally, like I said, brain tumor, two most common primary brain tumors. We look at primary brain tumors, glioma and meningioma. All right, glioma, also known as glioblastoma multiforme, GBM in short, or the source of tumor is not the nerve cell. Nerve cell is a neuron. Neuron usually, I think, not a cancer or something like that, but the glial cell that is around the neuron. All right. I wasn't able to extract a photo to show you, so that's why I just leave it blank. Right, and supporting and protecting the nerve cell. All right, the glial cell is the glial cell that surround it. Okay, it accounts for 60% of all adult brain tumors. So most of the time when an adult that have brain tumor, usually is glioma because it's 60% of it. All right. It is classified as a central nervous system because the brain is part of the central nervous system. Okay. Right. There are generally 4 stages. Right. According to its aggressiveness and recurrence. Like I said just now, stage 1 is a slow-growing benign tumor. Usually, if you have headache, constant headache, and then the doctor diagnosed already, say there is a tumor, and if you feel that it is not as aggressive and doesn't recur as oral, you will consider stage 1. So you're benign, they will tell you, observe first, come back and check again 1 year later, or if you feel something more serious. Stage 2, also same thing, but stage 2, you will start to have more of the reaction, like start to have severe, maybe blind, double vision, severe headache, the limb pain, all that kind of thing, right? A bit nauseating, more neurological problem. And at this stage, doctor will still say that it's benign, but if you want to go away, treat away with this all these symptoms, they'll say go for surgery, gamma, under gamma knife or something like that, for treatment, all right? And stage 3, once it progresses there, that means it's already considered cancer and it's aggressive. It can progress to stage 4 straight away, right? Very fast growing, and further treatment is a little bit difficult. And it will probably spread to other part of your body. Right? Next slide. Right. The other one is meningioma. Like I said just now, it is around the membrane of the brain. So here I extract a photo of it. It's around the membrane, around the skull area, near the skull. And that accounts for 20% of primary brain tumors in adults. All right. Generally, the meninges are what? How you have 3 layers. Usually it occurs in the center layer. Then it will grow from there. Then you press on your brain. Then probably it gets a bit too much. Then you start to lose memory, blur vision, that kind of thing. So, as it is, grade 1 is benign, grade 1 and 3 is still malignant, grade 2 is between the two. Then 90%, like I said, of meningioma are benign, right? So you press on your nerve until you have constant headache, double vision, maybe you have limb pain, nerve pain here and there. When your nerve is painful, a lot of area is painful all over your body, right? So same thing once it's considered to be stage 3, the final stage, any treatment is poor because of the blood-brain barrier. Very hard to have drug to get into the brain, right? So the treatment options are limited. That's why more and more doctors are looking into immunotherapy, right? Okay, so that was a little bit basic on the brain tumor. Those who want to know more, same thing, you go and read a little bit more on it. Okay, let's look at the first research report, right? On Lingzhi immunomodulatory effect, the first report. The first report was done by researchers from the Fujian Medical University, right? and in conjunction with Fujian Institute of Neurosurgery, Fujian Agriculture and Forestry Universities, all right? They did an animal research and they found that the polysaccharides extracted from Lingzhi, okay, fruiting body, can exert anti-brain tumor effects in the body, right? The extracted Lingzhi contains 6.49% of polysaccharide protein. So if I'm not wrong, our YK has something like that. Oh, right. The study, of course, in all animal study, they will have to implant the cells into the brain of the rats to induce brain tumor, right? And 1 week later, then they start to inject 50, 100, and 200 milligram per kilo of Lingzhi polysaccharide extract daily for 2 weeks. All right, so in total, it's about 3 weeks. First week, they inject to let the brain tumor grow in the brain of the rats. After that, then they inject Lingzhi polysaccharide to see how the treatment goes. Right? After 2 weeks of treatment, the tumor size was measured via MRI, and thereafter the experimental mice will continue to be fed till the end of their life. Right? That is roughly the research process. We will not go into very detail on it, but this is just the process. Right? Right, next, let's look at the result. Right? The result. Okay, after 2 weeks of treatment, the tumor size of the mice given 50 or 100 milligram of polysaccharide was reduced by 1/3 on average when compared with the untreated control group. So, like I said just now, they inject the glioma cells into the brain of the rats. The control group that was not treated, the tumor grew in size. Then after 2 weeks, when they treat them with tumor, and at the end of the 3 weeks, all in, they find out that the tumor size of the polysaccharide group, especially the 50 or 100, was only 1/3 on average. Right? Smaller than if you have no treatment now. Then also, they look at the survival time. Remember, I was telling you, after treatment, 2 weeks of treatment, then they leave them, continue to feed them and let them live and see how much longer that they live, right? They find out that 100 milligram per kilo, the medium group, live the longest. It averaged 32 days. Although one of the rats lived up to 45 days. The other two groups averaged about 27 days, right? The survival time of the 100 milligram group is the 1/3 longer than the average of 24 days of the control group. So if you look at the chart on the right, right, A, B, C, A is showing the tumor size, right? The tumor size of the in the red between the control and the 3 groups of Lingzhi consumption. B is the tumor size. You can see that the control, the left one, the left bar chart is 150, more than 150. The 50 is lower, 100. The 100 milligram is slightly around more than 100, whereas the 200 is back to 150. All right. Okay, then the survival, the C is the survival time. The red line at the very most is a control. They live the shortest, right? They live the shortest. But if you see the 101, some of them stretch up to 45. All right, so on average, as I say here, the average of the 100 milligram per kilo group was 32 days as compared to the control group, which is 24 days. So that is another 8 days more, right? 8 days out of 24 days is how many? Is 1/3, correct? 3 times 8 is 24, right? So it's 1/3 more. It's just like, you know, I don't know whether your there was a WhatsApp circulation of someone with cancer treatment that he spent $1 million for only 2 months, you know, of treatment, right? This is an example, right? Okay, next slides. All right, given the results of this experiment, the researcher want to know what happened, what kind of mechanism of action, how it works, why is the result different? Okay, further analysis of the tumor tissue. They cut out the tumor, then they look at the tissue. They found that the cytotoxic B cell, which is the CD8 plus, and other beneficial anti-tumor cytokine, anti-tumor cells, like interleukin-2, TNF alpha, interferon gamma. TNF is a tumor necrosis factor alpha. All right. Interferon gamma, these are all name of the cell. For us, we don't need to know so much, but just to know that that's the case. Were much higher in the Lingzhi group than the control group. So within, why is it that the Lingzhi group has a higher survival rate? Because they have more killer cells than the control group that is not treated, right? So partly explain the boosting of the immune system by Lingzhi, right? The study also found out that the splenic natural killer cells, which enhances immunity, were higher in the GLP, the polysaccharide group. So what does it tell you? It tells you that Lingzhi improve the killer cells. Lingzhi improve the immunity. As a result, it can fight the cancer. It can kill the cancer cells. All right. So it produces all these antiviral cytokines like interferon gamma, all that. So it is through all these immune system that gets into the brain, gets past the triple B barrier, the blood-brain barrier, to solve the problem, to kill the cancer cell, right? That's why they came out to the conclusion that Lingzhi helps to suppress the cancer. Okay, right. Okay, let's look at the next research report. Okay, before we look at the next report, we look at a summary of the study. All right, so the researcher determined that polysaccharide from Lingzhi was able to slow down the rate of growth of the tumor through activating the immune system I just mentioned, right? The immune cells of the mice to fight against the invasion of the malignant brain tumor cells, and thereby prolonging the survival period. I just mentioned about it lah. Right, the physiological structure of the blood-brain barrier of the brain did not prevent the effect of polysaccharide in inhibiting malignant brain tumor. Right? I mean, Lingzhi, I mean, you think that you take Lingzhi, how Lingzhi can reach the brain cancer cell, right? It reached the brain cancer cell through the immune system. It did not reach the brain cancer cell of the powder that you see in Lingzhi, right? It reached the cancer cells through activating, this powder activate your immune system to fight the cancer. So the experimental results show that of the 3 dosage, right, the medium dosage of 100 milligram per kilo has the best anti-cancer and life-prolonging effect. What could be the reason? It could be due to the modulating effect of polysaccharide, which is about regulating the immune system. So, and the level of state in the body rather than the dosage. So if the state of the body is weak, then you take more. If the state of the body is average, then this is 100. So it doesn't mean that the more the better, all right? Doesn't mean that, oh, in order to fight cancer, you have to take much more, right? So we take a modulating amount to just activate the immune system. Too much, maybe your immune system might just override and cancel off itself. It doesn't work anymore. Okay? Right. Let's look at the next report. Okay, next report is on Lingzhi protein that inhibits glioma. Okay? So an immunomodulatory protein extracted from one species of Lingzhi called Ganoderma microsporum. In short, they put it GMI, Ganoderma microsporum immunomodulatory protein. In short, they call it GMI lah. Was previously found to be effective in inhibiting cell migration and invasion in lung cancer cell. There was an earlier research report. Okay, you want to know about it, then you go to it. They refer the reference and show you to the lung cancer. All right. So there was a research report that shows that this GMI can inhibit lung cancer cell migration and invasion. And as you know, 50% of lung cancer metastasize to brain cancer. All right, since it can inhibit lung cancer cells, maybe it can inhibit the brain tumor cells. That's what the researchers thought. So this researcher from Taiwan, they use bioinformatics. Nowadays, a lot of drug research, a lot of research in this area use computer, all the big data thing, right? Bioinformatics is all the big data. Explore if the protein is also effective in reducing the invasiveness of GBM, glioblastoma, multiform, right? Cells, and improve the effectiveness of chemotherapy drug, temozolomide. Actually, talking about this bioinformatics, computerized drug discovery. You know, recently all those vaccine, all this is also basically using computer thing to do it, and they jump the gun before they do any red research. They use the computer to come up with a result. then they say it's effective. Oh, so that is the case here. Right. So GMI, GMB, GBM, sorry, is considered an aggressive brain tumor because it's already when it's classified as glioma, blastoma, multiform, it's already stage 3, stage 4. Okay. So it is a metastasis. It is aggressive and it really can spread to other parts of the body. All right, and chances of survival is very poor. Usually, one with brain cancer lives for less than a year or two. That's all. All right. The study found that GMI, whether used alone or in combination with this temozolomide chemo drug, significantly reduced cell migration and the invasiveness of glioblastoma multiform cell. Right? It inhibited 2 types of human GBM cell. Right? They are basically here in this example, in this study, they use 2 types of GBM cells called GBM 8401 and T98G. Right? 2 types. Because not all cancers are the same. Right? So they have extracted 2 types. They get the cells and they test it. Right, next slide. Right, so because this research report was not available, they only show a very limited amount. So therefore, we can only go through a little bit on the mechanism of action. Right? Like I said, it used a computerized study. So they had this heat map profile, so-called like this MRI thing, where you see the different color, that kind of thing. Okay, so this heat map profile show that GMI treated cancer cells were distinctly clustered, right? Were all grouped together, indicating that the cancer cells were prevented from further spreading to the surrounding tissue. Right, that was a result that they found, that they find that after the treatment, when they do it, this is a in vitro treatment. All right, this is not a animal study. This is a cell study. Right, they found that when they add this GMI, the cancer cell tend to cluster together. Right. The reason they gave was that it was able to reduce the slug protein, SLUG protein. And this protein is found in the cancer cell, responsible for its proliferation and drug resistance. Right? This protein causes the cancer cell to spread and causes the cancer cell to be resistant to chemo drug. All right. Since this experiment of GMI are able to cluster it together at a dosage of 0.2 micron millimeter or something like that, after 3 hours of treatment, right? The slug protein has been reduced in that 2 type of cancer cell that was tested as compared to control. So naturally, the conclusion of the researcher is that it reduced the migratory ability and invasiveness of the cancer cells significantly. Because once it's clustered, it doesn't run around, right? It group them up, it tie them up, and then so-called don't cause them to spread anymore. And then hopefully through the immune system, it will kill the cancer cell, right? So GMI, by targeting on the slug protein, not only dealt with the increase in cancer cell, right, but also improved the sensitivity of GMI to chemo drug treatment. So GMI, by targeting on the slug protein, right, not only dealt with the increase in cancer cell, right, it stopped the cancer cell from increasing, right, it also improved the sensitivity of GBM to chemo drug treatment. However, like they say, further in vivo studies need to be done. This is an in vitro study. This is a cell study. So they need to do a study on an animal or human to confirm this observation. All right. Okay. Next study. Right. Lingzhi suppresses meningioma. Okay. This study made by people from the U.S. Medical University of South Carolina and the Hollins Cancer Center. They found that triptanoid, especially ganodermic acid A and DM, that were extracted from Lingzhi, can inhibit growth of high-grade meningioma and prolong the survival time of tumor-bearing mice. High-grade meningioma, that means it's already considered stage 3 lah. And Lingzhi has over 300 triptanoid compounds. So A and DM is 2 of it. All right. So same thing, this one done an animal study. They implant human meningioma cells, IOMMB cells, into the meninges of immunodeficient elderly to establish an animal model. Same thing, this research report is because it is a protected study, so we are not able to access much more detail, but just the abstract, right? After 2 weeks, 10 milligrams per kilo ganodermic acid A or DM, right? They have 2 lah. One is treated with ganodermic acid A, one treated with ganodermic acid DM lah. Both have the same result, right? There was intravenously injected to the mice, and the treatment continued for another 14 days, while another batch were left to live out their life without further treatment. So this one basically to compare the survival time. The results show that the size of the meningioma in both ganodermic acid A and DM was smaller than the control group by about 60%, right? And the average lifespan of the control group was less than 30 weeks, while that of the Lingzhi group was more than 50 weeks. That means it is how much more? 60% more, right? So that's because we can only look at the summary. We are not able to look at the detail of it because it's a protected report. You have to pay for it to get it, right? So what is the mechanism? According to the researchers, what is the mechanism of action? Next slide, Jindong. Jindong, next slide. The mechanism of action of this ganodermic acid A and DM. How they come to cause the tumor to shrink and the people to the rats to live longer, right? They found that the liver enzyme, right, of the 3 groups were maintained at a similar level. First, why they look at the liver enzyme? Because it shows that it has no uh, toxicity against the liver, right? And it is protective and no detrimental effect to the neurons and the nearby brain cells. Okay, so that's why they want to look at the liver enzyme to show that it's not toxic to the body or even to the brain cells. Okay, liver is the organ in our body to detoxify whatever that come in. All right, so that's why they look at the liver enzyme. Okay, then they analyze the mechanism of action in the ganoderma acid. They found out that these two acids, ganoderma acid or DM, increase the anti-cancer cell protein, NDRG2, the anti-cancer cell protein called NDRG2. And at the same time, reduce the number of anti-apoptotic protein, Ki67, in the tumor tissues. Imagine they go into these nitty-gritty cells of the body, all right? This, what is this? Anti-apoptotic protein. What's apoptotic? Apoptotic is killing the cells, right? So they found that you reduce all this, so as a result, your tumor can reduce. Okay, the NDRG2 protein is said to be the main enzyme that affect the development, deterioration, and recurrence of meningiomas. Right? In meningioma, in malignant meningioma, such enzymes were found to be insufficient. So these two ganoderma acids were able to increase this protein. So when you're able to increase this protein, so as a result, it can go about to suppress the cancer cell from growing and killing it and making bigger. Okay, so therefore their conclusion of the study was that ganoderma acid A and DM, or in short terpenoid, increase this NDRG2 protein. And it was the key, because they look at this protein, they find out that the protein in the rats that was treated were higher. So, and this NDRG2 protein was the one that can reduce the cancer cell. So, as a result, it increases cancer cell death and reduces the cancer cell from proliferating, spreading. Okay, a very technical and very detailed, which is also, I also find it honestly very hard to explain, right? But imagine the researcher go into the detail of the cell to explain why it has been rejected, right? Why it's able to kill the cancer cells and prolong life, right? Okay, come to the conclusion. Come to the conclusion of this research report, right? What has been seen as a despair, right? I mean, one who has been stuck with a mother of all illnesses, right, can now at least see hope. I remember I just opening say that the chances of survival, the prognosis is kind of like for 2 years it's only like water only. So these 3 studies shows that there is a possibility that Lingzhi can help. to inhibit the tumor from growing, right? At least it gives the patients a chance to prolong their life, right? Instead of that kind of prognosis as given, which is 27% for less than 2 years. Okay. So, as for brain tumor that spread from other parts of the body, characteristics of their cancer cells are also very similar. Although these studies was based primarily on the primary source, right? But the characteristics of the other brain cancer are also the same. So, as it is, Lingzhi polysaccharide, Lingzhi tripeptide, Lingzhi protein will all have good direct or auxiliary therapeutic effect, right? Therefore, no matter whether the tumor is primary or secondary, malignant or benign, the key to prolonging life and improving quality of life in the face of a dreaded disease is to consume more Lingzhi with all its effective components. I say, give life a chance, give Lingzhi a chance, and Lingzhi will give you a chance to live longer. Okay? Right, thank you. That's all I can share about it.